Thank you for introducing me. And my name is Ryu-ichiro Tanaka. I'm with Tokyo Women's Medical University. Okay. Today I'll be talking about suspension culture of bovine myogenic cells with microcarriers using edible basal medium and bovine serum. So I will explain about background. Okay. And we aim to create cultured meat with myotube with likely to real beef. And to create this next generation cultured meat, we need expansion culture technology for myogenic cells. So we have been developing a high efficiency expansion culture technology. And to create cultured meat with myotube, a large amount of myogenic cells which can differentiate to myotubes are needed. So we know that the suspension culture can produce more efficient than plate culture. And however, the suspension culture is difficult to control culture condition and to maintain the differentiation capacity of myogenic cells. So this is a gap between plate culture and suspension culture. Therefore, in this study, we have developed a suspension culture method enabled to maintain differentiation capacity of myogenic cells. By the way, the price of serum is a major problem of cultured meat production. So therefore, instead of FBS, we used adult bovine serum because adult bovine broth was byproduct of conventional meat production. So we can get cheaper than FES. The graph on the right shows the result of comparing FES and bovine adult serum in plate culture. So the MEM supplemented with 10% FES and 10% bovine adult serum was used. Although the proliferation rate was lower than FES, but it can grow bovine myogenic cell by adult bovine serum. So next, I'll talk about methods. We used a 30 milliliter bioreactor for this experiment, and we used Syncymax-2 microcarriers. The microcarriers were coated with I-matrix 511, which is laminin fragment for enhancing adhesion and for maintaining differentiation capacity. So in our method, edible basal medium, which made by integrated culture and called IMM, was used. And the component of amino acids and vitamin and so on are same to the MEM. So instead of FBS, we used adult bovine serum. And bovine myogenic cells were collected by the previous reported method, and collected myogenic cells were seeded with IMM supplemented with 10% bovine adult serum in the 30 milliliter bioreactor. And the concentration of cells was 40,000 cells per milliliter. And after static culture in 37-degree incubator for 2 hours, and the bioreactor was rotated at 50 RPM. So during suspension culture, 50 milliliter medium was changed every 2 days. And the proliferated cells were collected by trypsinization every 4 days or 6 days. And some collected cells were seeded again and with microcarriers in bioreactor. Some collected cells were differentiated on the dishes for the assessment differentiation capacity. So I'd like to show the result of proliferation rate. So the left graph shows the proliferation rate of 4 days or 6 days interval passage in culture. The proliferation rate of 4 days interval passage in culture tend to be higher than 6 days interval. So by culturing for 6 days, there is no further proliferation. So the right graph shows the cumulative proliferation. Over 24 days passage culture, in the case of 4 days interval was 110,000-fold, and in the case of 6 days interval culture was 2.4 thousand-fold. So 4 days interval was markedly higher than 6-day interval passaging culture. So we checked the surface of the microcarrier after culturing. And F-actin dyed green by phylogen solution and nuclei dyed blue by Heckist solution. And, and compared to 4 days, some 6 days microcarrier were aggregated. So the myogenic cells on this microcarrier were overconfluent. So from this, we considered that the proliferation rate didn't become high from more than 4 days culture. And in addition, myogenic cells might different to myotube and under overconfluent. So for this reason, the following results were obtained. The collected myogenic cells were seeded on culture dishes and differentiated to myotube every passaging. So the myotube was dyed green by desmin antibody. And this figure shows the dyed myotube in both 4 days and 6 days interval passaging culture. Myotube were decreased, as you can see. So we compare the relative area of desmin positive, and this graph shows the relative area in case of 6 days interval. The differentiation capacity was significantly decreased at passage 2. But in case of the 4 days interval culture, the differentiation capacity was significantly decreased at passage 5. So therefore, it can be said that the 4-day culture maintains the differentiation capacity more because of passaging before being overconfluent. So next, we performed the experiment using basic fibroglass factor. So we know the basic FGF enhances proliferation and regulates differentiation. So we added the basic FGF at a concentration of 5 nanogram per milliliter. So we tried the same experiment comparing 4 days and 6 days interval passaging. Okay, so the result of the proliferation rate were as follows. And in case of 4 days interval, the proliferation rate was higher than without basic FGF initially, but it went down significantly. So in case of 6 days interval, the proliferation rate was similar to without basic FGF. So the cumulative proliferation rate over 24 days were no markedly different to without basic FGF. So same as before, we checked the surface of microcarriers because of enhancing proliferation rate by basic FGF. The microcarriers of 4 days culture were aggregated. So I think that this condition caused significantly decrease in the proliferation. So same as the case of without basic FGF, so the collected myogenic cells were seeded on culture dishes and differentiated to myotube every passaging. In case of 6 days interval passaging, differentiation capacity was markedly decreased as same as the case of without basic FGF. So differentiation capacity was compared as same. And same in the case of without basic FGF. The differentiation capacity was significantly lower in the 6-day passage. So on the other hand, the differentiation capacity was maintained in the 4-day passage with a significant decrease at passage 6. So this graph shows the relationship between the number of division and the relative desmin positive area. So 6 days interval passaging culture showed a marked decrease in differentiation capacity relative to the number of divisions. And while 4 days culture tend to maintain differentiation capacity relative to number of division, and which was further maintained with the addition of basic FGF. So the expression of PAC7 in myogenic cells collected at passage was also compared by immunostaining. And the percentage of PAX7 positive cells is shown in the graph. And the same with the differentiation capacity, the percentage of PAX7 positive cells tend to be higher in the 4-day interval passaging. So it also tended to be higher with basic FGF was added. And so how much weight did the cell obtain by our method over a 24-day culture period? But it's just calculating. So. And a previous paper from our laboratory reported that 10 million cells were collected from 2 grams of bovine cheek muscles. So these myogenic cells expanded to 10 billion cells through suspension culture over 24 days. And 10 billion cells weighed about 3.5 kilograms. And this demonstration was conducted on a small scale of 30 milliliter. But if large-scale expanded culture can be achieved, 1,750 folds the initial 2 grams could be produced over 24 days period. Okay, this is the last slide. I'd like to finish with a summary. Our question is, what are the culture conditions that can maintain the differentiation capacity of bovine myogenic cells using microcarriers in suspension culture? So our conclusion is simple in this study, and similar to plate culture, it is important to take cell density into account in passaging culture. So compared to 6-day interval passaging, 4-day interval passaging is maintaining differentiation capacity and faster proliferation. And in terms of future perspective, we are considering that the scaling up of culture over limiting it to liter scale. And additionally, we are working on the development of edible or reusable microcarriers to promote sustainable production. So we think that the simple our method will contribute to produce cultured meat with myotube, which is next generation cultured meat. Okay, thank you for your attention.
Thank you so much. Questions?
Thanks for the very nice presentation. Did you also try using the basal medium, which is ISCCM, with reducing serum concentration?
Yes, concentration.
With 0% FPS?
No, I did not try it.
Okay.
Okay.
Thank you for your talk. I see that your differentiation images look really good.
Okay, thank you.
I was wondering how you differentiated, like for example, what media and how you stopped them from lifting off, since they differentiate so well, and they usually can pull themselves off the plate.
Okay, I use Maastricht 511 coated dishes, and I seeded collected cells and near confluent, and 1 day after, we changed to differentiation medium, which is DMM supplemented with 2% FPS, and 3 days culture, and we okay. So 3 days culture. Thank you.
So I also have a question for you. So did you add microcarriers during this passaging? Because you said that you changed media every 2 days, and then you did 4 or 6 day passaging. You also added new microcarriers to the spinner flasks during this?
Yes, I use in this experiment. I use new microcarriers for every passage.
So for the passages?
Yes.
Okay. Yeah.
Yes.
Thank you.
Thank you.